Good morning. I hope you're doing well. It's Tuesday morning here, whenever you watch this, whatever day that will be for you. I start with this slide just in anticipation of our first video conference, if I get brave enough to try one of those. And my hat today is green for spring, and this is in my front yard this morning, and so I just thought I'd spread a little sunshine your way. I think I left you, I was at the bar drinking my unsweet iced tea with, who was it? It was Gerund and Infinitive, I believe. And so, and then two quotation marks like walked into the bar, and we were having a great time and yucking it up. But then when oxymoron walked in, the silence was deafening. This is so good, I can hardly stand it, and you probably feel the same way. So let's get on with today's lesson, which is going to be at least carbohydrates and lipids, and maybe proteins, depending on how I get close to my 512 megabyte upload limit for Schoology. So here we go. So I'm going to start again with the learning objectives are keys, right? I got a little gold paint for you today. The learning objectives are keys because every single question on the test, again, no matter what kind it is, has to be based on at least one learning objective, and they will use the language like describe and monomers and macromolecules. They'll use the language of the learning objective, so they're always good to pay attention to those and kind of start with those. So this tells you what you need to know about this particular learning objective. And so here we have hydrolysis and dehydration synthesis. It says, describe the properties of the monomers and the types of bonds. Well, here they tell you the type of bond is covalent bond. That's a pretty simple part, right? But then you also pay good attention to these because these tell you what you don't need to know beyond the scope of the AP exam, and that is specific nucleotide, amino acid structure, glycine versus proline, for instance. So that's where we're starting. So here we go with dehydration synthesis and hydrolysis. And remember, those are the words that they used in that last slide there. So here we have the idea that to put two things together, one of them gives up, yellow isn't too good to see here. I'm going back to garnet. One of them gives up a hydrogen, one of them gives up a hydroxide, and that's where the water comes from, which is why it's called dehydration. This is endergonic, and we'll get to enzymes later because that catalyzes all this stuff and makes it happen faster. So here we have the opposite, right? Hydrolysis. This is what's happening to your lunch, right? This is also sometimes called digestion. This is one way that molecules can break apart, and this is the way that all the organics are broken apart, along again with water broken apart to its constituent hydrogen and hydroxide. So here we go with carbohydrates. Complex carbohydrates comprise sugar monomers. What they wanted to say here was that complex carbohydrates are comprised of sugar monomers stuck together by dehydration synthesis. We'll forgive them for that. They should have had me proofread this first. Here, carbohydrates comprise linear chains of sugar monomer. I would not word this way, but these again are the words of the syllabus, right? And linear or branched, we'll look at that real fast. That's pretty simple. All of this hopefully is, again, lots of review stuff. So we have monosaccharides, disaccharides, polysaccharides. They don't make their way into the syllabus, but of course they're common kind of words here. And here we have an example of sugar monomers whose structures determine the properties and function of these complex carbohydrates, also known as polysaccharides. That means you have different monomers. You don't need to know the structure of each one, but just the fact that there are different ones. And if this polysaccharide is made of this one, it's got these properties. That one, it's got those properties. We'll come back to that later also. All right. Isn't marketing wonderful? Because doesn't make this, what's it make, doesn't this make you want to buy this because it's carbon free. And not only that, it's certified carbon free. And if you don't know anything about science, this might cause you to think that this is like better than sugar that's like not organic, which there's no kinds of sugar that aren't organic. This costs 3 times as much as just a regular sack of Dixie Crystal sugar. Here we go. So here we have just a specific example, right, of these two monomers being stuck together to make that organic, right, but not carbon free. Interesting there. Sugar, here we have the dehydration synthesis part. So polysaccharides, again, also named in the syllabus as complex carbs, right? So don't call glucose a complex carb. That means several of them stuck together. If their function is energy storage, we call them starches, ST for starch, ST for energy storage, right? And we have some that are just linear. And in these starches, they also often helix up, just like we're going to get to proteins in a while, and DNA, obviously. And then sometimes, again, they're branched. So we have these starch molecules made by plants, made by animals, made by fungus and other things too. But again, it's the nature of the branching and things that makes different kinds of these. And that's about all we need to say about those carbohydrates. So here we have what I think is probably where they would go about a change in the subunits of a polymer that may lead to changes in structure or function of a macromolecule. I really think that where this is talking more about proteins and nucleic acids, which we'll get to soon, right? Because the change in the subunit is not something that happens in a carbohydrate. It doesn't like mutate so that it has different subunits like a nucleic acid would, that would then lead to a protein having different amino acids. So I think this is more about those. But here we have sugar molecules whose structure, I think this one might be more about alpha and beta glucose, because you don't find complex carbohydrates being made of a bunch of fructose, for instance. They're just glucose polymers. But whether they're alpha or beta glucose polymers makes a big difference in their properties and functions. So here's our example. These starches are alpha glucose, right? And this allows enzymes, which have evolved, enzymes to break them down easily and be used for energy. And cellulose, however, is made up of beta glucoses, and that causes them to not be able to be broken down easily, which is exactly what you want in some kind of structural strength, strong building block molecule. So when you build these polymers with beta glucose, that puts the OH groups alternatingly, I didn't do a good job of that, but alternatingly in a way that they can hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond with other ones to make long chains. It's not that early. To make long chains and make this very strong building block for most notably cell walls of plants. Okay, so now we're to lipids. Okay, now here we have the essential knowledge about lipids. They are non-polar. Differences in saturation, right? They want you to know that. Determine the structure and function. Always the structure relates to the function, right? We have phospholipids focused on for their role in cell membrane activity, and they have polar regions that we call hydrophilic and non-polar regions that we call hydrophobic, right? Why they didn't put hydrophilic in here, I'm not sure. But the molecular structure of specific lipids is again beyond the scope. So here we have your typical kind of lipid that's made of a fatty acid chain. We have here again the saturated versus unsaturated idea. It's the fatty acid part where that comes into play, and the fact that there are no double bonds between carbons here leads to the saturatedness of hydrogens, which is what is the saturated part of that. And so this is a saturated fatty acid. It's straight. And here's glycerol. It doesn't get much attention here. But if you have 3 saturated fatty acids, they can be real close to each other, and this lipid kind of is packed in with other lipids. And that's what makes an unsaturated fatty acid different in structure. This phospholipid coming up is composed of, as you can see, an unsaturated fatty acid, and this causes phospholipids to not get too close to each other when they make up a cell membrane, which causes it to be fluid, which is a lot of part of its function. This is social distancing at the molecular level. That thought just came to me, so isn't that nice? So here we have again the hydrophilic, hydrophobic, and to get those words straight and keep those words straight and the parts of this molecule, this is where I think again they ask you the components of these, the nature of the components of these big molecules. This might be part of that. Here's that other kind of lipid which they don't mention, so I'm only going to throw it in here real fast, that lipids basically are insoluble in water, and that's the only reason we humans put these kind of molecules in the same class as ones that look like those other ones with the fatty acids, because these molecules don't care what we call them, but we call these lipids as well. Even though they have quite different functions, these are very common signaling molecules, but they also, cholesterol is a difference between cell membranes in plant cells and animal cells, and so that connects later in the syllabus.